Hi, everybody. Remember last time we went over some basics of taxonomy. I talked a little bit about what it is, how it works, a little bit of the history, and we went over a couple of examples. And that should give you enough background to dive right into the phylogenetics and systematics portion of it. So remember in this Prezi, which is available on Blackboard, we worked mainly in this area at first. Now we're going to go into here. So let's zoom in. First of all, let's spend a little bit of time defining some terms here. Phylogenetics. Okay, so phylogenetics is simply the evolutionary history of a species or group of species. And now, when we talk about this, we'll often use a picture to display the evolutionary relationships, and this is called a phylogenetic tree. The other word I'd like to define right up front here is systematics. Systematics is simply the process of classifying organisms according to their phylogeny. In other words, according to their evolutionary history. And we'll practice doing this in a number of different ways. We will use some morphological characteristics. We will also do a case study on anoles in the Caribbean, and we'll use molecular and genetic techniques. But some people use fossil evidence to construct phylogeny. So there's lots of different methods of doing this, and all of these methods sort of fall under the umbrella term of systematics. Okay, let's continue to define some more terms. I have to use these words when we talk about evolutionary relationships, these words primitive and derived characteristics. So first of all, primitive characters are characters that originated in the ancestor of the taxon. Derived characters, in contrast, are characters that are unique to the taxon and distinguish it from its ancestors. Okay, so let's go back to this example that we looked at in the phylum Chordata. Remember, all of these organisms in this phylum share several primitive characteristics. They all have dorsal hollow nerve cords, for example. We talked about how a derived characteristic of mammals that distinguishes it from the rest of the chordates is hair. Okay, so let's go to a concept check here. I already gave you one, but relative to other chordates, which of these characters is a derived character of mammals? Is it cellular respiration, lactation, a dorsal hollow nerve cord, or all of the above? Now, if you answered lactation, you're correct. Lactation is something that distinguishes mammals from the other chordates. Okay, so it's a derived character. Let's go to the next one. And this question is for all mammals, which of the following is a primitive characteristic? Is it cellular respiration, lactation, or a dorsal hollow nerve cord, or all of the above? Now here it gets a little tricky. All of the above are primitive characters of mammals. And the question to ask yourself is, did the common ancestor of all mammals have these characteristics? And the answer is yes, they all use cellular respiration, they all lactate, and they all have a dorsal hollow nerve cord. Now characters 1 and 3 are primitive characters of other groups as well. So we'll practice this in class. It is confusing. This is just a little taste. All right? When constructing phylogenetic trees, we'll often use this principle of maximum parsimony. And what parsimony is, what this principle says, is that the tree that requires the fewest evolutionary events is the most likely. Let's use a molecular example to illustrate this. Here we have 3 species of birds. There are 3 phylogenetic hypotheses possible for these three species. We could have number 3 being sort of the outgroup and numbers 1 and 2 being sister taxa. In other words, they diverged from group 3. We could have species 1 and 3 being sister taxa with 2 as the outgroup, or we could have species 3 and 2 being sister taxa, the most derived taxa, and number 1 being the outgroup. And in order to get at which one of these is most likely, we can look at all kinds of things. We could look at colors, we could look at beak shape, we could look at tail length, we could look at any host of morphological features, but that often results in categorizing or classifying organisms by analogous traits. A more objective approach would be to use nucleotide sequences. Okay, so I'm going to do my best here to explain this. We might need to come back to this in class because I don't want to take too much time on it right now. But let's imagine that we have these 4 nucleotide sequences, and we know that the common ancestor of these 3 species had the following sequence, A, G, T, T. Now, if we take the same region of the gene for the 3 species that we're interested in, we can compare those to the ancestral sequence, and we can look at which of the 3 hypotheses requires the most changes in nucleotide sequences. Now, your book goes over this a little bit in chapter 20, so I encourage you to look at that. We are going to use some software in class to look at the annuls that I talked about earlier. But let's go through this. Let's see how it goes. Okay, so let's look at this first site. Species 3 has the same nucleotide at site 1 as its ancestor. Species 1 and 2 have that site changed to a different nucleotide. And so that is what this first phylogenetic tree is illustrating here. Okay, we have at site 1, we have 1 change to C, okay, with the outgroup here being species 3. Now, if you look at the other 2 hypotheses here, the other 2 phylogenetic trees, you can see that this one, the one that has species 1 and 3 being sister species with 2 as being the outgroup, this tree requires 2 separate changes. It requires an independent change from A to C for species 2 and an independent change of A to C for species 1. And you can see this phylogenetic hypothesis requires 2 changes as well. And if you do this for all 4 sites, you can see that this hypothesis requires 6 different evolutionary events, and the other 2 hypotheses require 7 different evolutionary events. And so the principle of maximum parsimony would say, okay, since this hypothesis requires the fewest number of evolutionary changes, it's probably the most correct. All right, so forward with defining terms. Your book spends a little bit of time on this, so I want to spend a little bit as well. A clade is a group of organisms that includes an ancestral species and all of its descendants. And this is often synonymous with a monophyletic group, and not all groups, not all taxonomic groups are monophyletic. Okay? This can be confusing. So again, let me do my best. We can come back to this in class. But here we have a phylogenetic tree, and we have different examples of monophyletic, polyphyletic, and paraphyletic groups. Okay, so this first group in the blue is an illustration of a real clade, a monophyletic group. And it's a monophyletic group because it contains the common ancestor and all of its descendants. If you move up here and you look at this group containing E, F, and G, this group in the yellow, this is a polyphyletic group. This group does not include the common ancestor of all of these organisms. Okay, so this is polyphyletic. A paraphyletic group, which is illustrated by the pink box here, includes the common ancestor and some, but not all, of the ancestor's descendants. So this is what you would call a paraphyletic group. Again, this can be confusing. Let's look at another phylogenetic tree and try to evaluate them. Which group is paraphyletic, which group is monophyletic, and which group is polyphyletic. So remember, paraphyletic is sort of an incomplete group. It contains the ancestors, or it contains the common ancestor, but not all of that ancestor's descendants. Okay, so which of these groups is paraphyletic? Take a second to think about this. If you said blue, you are correct. Okay. Now, which of these groups is monophyletic? Remember, a monophyletic group contains the common ancestor and all of its descendants. Okay, so the monophyletic group that is illustrated here is the yellow one. Notice that it contains the common ancestor and all of its descendants here. Okay, and finally, the polyphyletic group is the one that does not contain the common ancestor, and that's illustrated by the red. Okay, so let's come back to this in class if you need to. Feel free to come to lecture with questions. We can spend as much time on this as you want. I'm not going to dwell on it on the celebrations of knowledge or quizzes or anything like that, but I thought I'd go over it a little bit because your readings do. All right? All right, on to some practice. Okay, now you can look at this a little bit prior to class, but this is what we'll do when we begin the phylogenetics and taxonomy, the diversity portion of the course. We're going to spend a lot of time building trees in this class. I think that it's a good way to illustrate evolutionary relationships. It is, however, challenging. So this is what we'll do. We'll get all of the conceptual stuff out of the way, which is what we just did, and we'll spend time in class practicing. All right? Thanks for your attention.